Now we will discuss how we can detect this E. coli in drinking water. Although microbiological determination or enumeration of E. coli or any other pathogen is a very long step, it can be very complicated because there might be a lot of different steps involved for every different pathogen or their strain. We will discuss some of the generic procedures that can be used for detection of E. coli. The method that we are discussing here is most probable number that can enumerate the coliforms or fecal coliforms or aerogenic E. coli. By aerogenic, I mean gas producing E. coli in food, especially in water, that may be coming from any source, for example, the source from processing of any food as well. It does not enumerate the E. coli serotypes that are associated with human diseases or foodborne diseases, as we discussed. For example, this strain O157H7. So this method is not going to tell us about these particular serotype. We need to have more detailed analysis. For example, certain PCR techniques for letting us know about these other isolates or serotypes which are disease linked. But they can also this MPN number can give us a statistically probable number of pathogens or other E. coli that may be present inside the water. So this process, as most of the microbiology technique, it would involve multiple tube fermentation. We would be using many different test tubes in which the fermentation can take place by inoculating these tubes with the water that we need to determine E. coli in. And then they are given certain conditions and different growth medium, specific time and temperature by which we get to know that what are the indications of growth of different kind of E. coli or fecal E. coli within these tubes. And by looking at the most probable number table, we can look at the probability of presence of these pathogens in these food or drinking water sources. This method is basically progressive, by which I mean that we cannot actually determine, like any other chemical analysis, at once that how many number of these microorganisms are present, whether they are gas formers or whether they are disease causing. We only get to know that at first point we need to look at whether the E. coli is present inside the food or not, and then we look at whether there are any fecal coliforms present, and then the third step, we can confirm this presence of E. coli by other different tests depending upon if it is present or not. And then we use this statistical table, this most probable number, by which we get to know the possible amount of these microorganisms present inside the food. If we talk about, mostly we look at these expressions such as coliforms or fecal coliform, although there is no taxonomical validation of saying whether do we have a coliform or whether it's a fecal coliform or not. So taxonomically we cannot differentiate between the two, but they are only used as an indicative microorganisms, by which I mean that when we found that there is presence of E. coli or a fecal coliform within the food, it might give us certain information. For example, this information might be different possibilities, like your food has not been properly processed, or there might be a sound processing, but then later the food has been contaminated by these pathogens. So once we do this analysis of presence of E. coli or fecal coliform, and we get positive results, that would mean that probably the food was not properly processed, or it was not handled after processing, and it has been contaminated by these microorganisms. Another thing is the growth of microorganisms. For example, these fecal coliforms, if we have fecal coliforms, that may tell us that the food has been contaminated and the microbial growth is taking place within a specific food. So this is the only importance of indicative microorganisms, that they can let us know that there has been some problem in processing or handling of food, and that is why they are present in certain foods. So if we talk about this method, as we earlier discussed, we normally get 5 replicate aliquot samples to an extinction, by which I mean that we keep on diluting these tubes to a level where we don't find any pathogen to that level, and from that we get these MPN numbers, and they give us the end results. Gas production is one of the criteria that can define certain aerogenic E. coli, and this is done by the LST broth, that is Lauryl Sulfate Tryptose broth, that is used as a test medium. If the lactose can be fermented in this broth, and then gas production from this another medium which is called as BGLB, Brilliant Green Lactose Bile broth, if there is a gas production, that would confirm the presence of coliform. Gas production at 45 degrees C in E. coli broth is again used as confirmation of E. coli. Normally we look at the microscope and look if there is dark centered colonies. They might have a metallic sheen, or it might not be there, so they normally let us know that probably there is a fecal coliform present. And once we take these positive samples having fecal coliform apparently there, then they are streaked onto another medium which is called as L-EMB, or levine eosine methylene blue agar, that can then, if they show any of the sheen on this specific medium, then we say that fecal coliform is probably present. But again, we then need to do a lot of other biochemical tests to confirm these presence of these microorganisms. So it is not only an exhaustive process, there are another lot of biochemical processes that can actually confirm whether the coliforms or fecal coliforms are present or not, and what are their most probable number within the food. So this was a general microbiological step to determine or look into whether the E. coli is present within a food or not.